one.
Hello, my name is Paola Lopez, and I'll be discussing a journal article called Food Allergy, a review and update on epidemiology, pathogenesis, diagnosis, prevention, and management. Some background information about food allergies. Food allergies are defined as adverse health effects that stem from a specific immune response upon exposure to a specific food. This is different from food intolerance, which is defined as non-immune reactions that can include metabolic, toxic, pharmacologic, and undefined mechanisms. A large number of studies have been done over many, many years that has yielded in data that shows that food allergies are not only common, as they are present in about 10% of the world's population, but they have also been increasing over the past 2 to 3 decades, especially in children residing in industrialized or westernized regions. It's important to learn more about food allergies and how to diagnose, manage, and treat them because food allergies can actually be life threatening due to anaphylaxis. And as of yet, there is no true way to prevent or cure them since our understanding of this condition is still very, very limited. The objectives of this study was to review information that would allow people to get a better understanding of food allergies. This study also includes advances, updates in the areas of epidemiology, pathogenesis, diagnosis, and treatment between the years of 2014 and 2018. This review uses a multitude of studies and data that have been obtained in order to highlight genetic, epigenetic, and environmental risk factors that have come to light while also shedding light on potential prevention and treatment strategies that can help those at risk. This review will also delve into the pathophysiology of food allergies, diagnostic methods, and management of multiple food allergies. As I stated before, this is a review that includes a variety of studies. The studies that were named in this review include various populations with studies that have 1-year-olds in Australia, children 5 years and younger in about 89 different countries, those that are between the ages of 6 to 19 years old in the United States, and children in 2 urban tertiary care allergy clinics, and those in New York City public schools. There's a number of other studies that include a variety of populations, all falling within this age range. Some are divided by race, mainly focusing on those who are either non-Hispanic Black or non-Hispanic white. The types of studies you'll see here are cohort and cross-sectional studies that were used in order to determine the prevalence of food allergies, case control studies that were used in order to determine the risk factors associated with food allergies, and a variety of other studies, including survey studies, genome and epigenome-wide association studies, and practical clinical studies. These were used throughout in order to determine diagnostic methods, treatment methods, as well as the relationship of genetics and epigenetics to food allergies. Some examples of the sample sizes used are given throughout this slide, including a sample size of 38,480 individuals who were cited as using the United States Household Survey, the Europrevail birth cohort, which had 12,049 infants participating, the Australian Health Nut Study, which included 5,276 children, a study of public schools in Australia, which included a sample size of over 550,000 students, and a National Health and Nutrition Examination Survey, which included 7,896 participants. This goes to show that every study included in this entire paper had very large sample sizes in order to obtain the data that was used to support the hypothesis presented. Most of the recruiting strategies used were allowing people to self report via surveys, while some of the other strategies were to use students in public schools as the data would be easier to obtain. Others were patients at clinics. There doesn't seem to be any specific recruiting strategy that this review sought in the studies that were used, but it is important to note that majority of these studies looked for individuals that were between birth and the age of 19 years, specifically in industrialized and westernized countries. As previously mentioned, the inclusion criteria for the studies used in this review include people from the ages of birth to 19 years. All of the studies were conducted only in developed countries, and the majority of participants were either patients from clinics, students at public or government funded schools, or were surveyed through government sponsored surveys. It would appear that none of the studies in this review included participants older than 19. There were no undeveloped or third world countries included, and individuals with uncommon allergies, which are food allergies that are not included in the top 8, were also not included in these studies. A number of the studies that were included in this review were randomized studies that were used in order to determine prevalence, diagnosis, management, and possible future therapies and treatments. Studies that were included in this review that dealt with genetics and epigenetics, treatment and management of food allergies included controls versus variable groups. One of the examples of these is a case control study that evaluated 106 cases and 76 control subjects. This study measured DNA methylation at 485, 512 genomic loci. Upon studying this paper, you will see that the statistical analyses include relative risk and odds ratio, which were used in a variety of different studies that were presented in this review. The results that were determined by this review are that food allergies are in fact on the rise in developed countries and are more common in younger children than in adults. It also determined that the most common mechanism leading to various food allergies is the breakdown of immunologic and clinical tolerance to foods, resulting in IgE-mediated reactions or non-IgE mediated disorders, which would include eosinophilic esophagitis, food protein-induced enterocolitis syndrome, and or food protein-induced proctocolitis. They also determined that the most important diagnostic tool for food allergies is the patient's clinical history, and that the use of schematic diagnostic algorithms that are able to consider the history, epidemiology, pathophysiology, and test results, including identification of the trigger foods, would lead to better diagnosis. There's still a lot of work to be done in order to ensure the proper management of allergies in terms of product labeling and the availability of emergency medications in schools and childcare facilities, especially considering that majority of nannies, schools, daycare facilities all state that they are uncomfortable with administering emergency medication to children who are having an allergic response. The most promising prevention method is early introduction of common allergens to infants. The conclusion of this review states that in the 4 years since the initial review, there have been a great number of advances